Hello, and welcome to Anatomy 100, assignment number 1. I am Aaron Vivian, and I'm going to be taking you on a journey through the senses of smell and taste. Chemosensation, which is the sensing system that taste and smell belong to. Nerve endings on the mouth, throat, and nose identify the certain sensations which give us the ability to taste and smell, with the fundamental chemical and cellular sensory features, and go all the way to the organism and organ system level of these senses. Cellular and chemical characteristics of taste and smell. Our first stop on analyzing the sense of taste on the chemical and cellular level is to actually analyze the taste bud. Each cylindrical taste bud is composed of numerous taste receptors called gustatory cells. The dendritic endings of each gustatory cell is formed by a slender gustatory microvillus, sometimes called taste hairs. The gustatory microvillus extends through an opening in the taste bud, called the taste pore, to the surface of the tongue. This is the receptive portion of the cell. Within the oral cavity, saliva keeps the environment moist. Molecules in the food dissolve in the saliva and stimulate the gustatory microvilla. This classifies them as chemoreceptors, as food molecules must be dissolved in saliva before they can be tasted. The gustatory cells within the taste buds are specialized neuroepithelial cells that have 7 to 10 day lifespans. Population of stem cells, called the basal cells at the end, constantly replace these gustatory cells. There are 5 basic taste sensations for our tongue. Although there are thousands and thousands of different flavors that give uniqueness to our food, there are only 5 different taste sensations that we can distinguish. This is sweet, salty, sour, bitter, and umami, meaning delicious flavor in Japanese. Analyzing the sense of smell on a chemical and cellular level, the sense of smell is also known as olfaction, the process of smelling through dissolved molecules in the mucus in our nasal cavity. Although this sense in humans is much more underdeveloped compared to other species, humans can still distinguish a single odor amongst thousands of others. These odors are broken up into 8 primary odors and thousands of secondary odors. Primary odors are able to be distinguished by a majority of people. For example, mint is a common scent accepted by a large percentage of the population who have experienced its scent. Secondary odors are usually a combination of chemicals, making it difficult for a majority to distinguish all odors. For example, flowers can produce over 100 different odors. Our next stop on analyzing the sense of smell on a chemical and cellular level is to look at the olfactory receptor cell. This cell is located in the nasal cavity. The cell first catches molecules in the mucus layer, and then the olfactory hairs detect what the molecules are. And through the nervous system, it goes to the olfactory bulbs, and that determines what the sense of smell, or what type of smell that is. We now move on to tissues and organ characteristics of taste and smell. The olfactory epithelium is the specialized epithelial tissue inside the nasal cavity of the olfactory system, directly responsible for detecting odors. In our nasal cavity, the olfactory epithelium lines the superior region of the nasal cavity, including both the superior nasal conchae and the inferior surface of the cibriform plate. The cilia traps odor molecules as they pass across the epithelial surface. Information about the molecules is then transmitted from the receptors to the olfactory bulb in the brain. Olfactory organs. Found directly within the nasal cavity and composed of several components. However, this specialized epithelium consists of 3 distinct cell types: the olfactory receptor cells, which detect the odors, the supporting cells that enclose the olfactory neurons and sustain the receptors, and the basal cells that function as a neural system to continually replace the olfactory receptor cells. Olfactory organs have olfactory receptors that can detect stimuli in the air that we breathe. This is another image of the olfactory receptor cell in detail. Olfactory epithelium to connective tissue. Internal to the olfactory epithelium is an areolar connective tissue layer called the laminar propria. Within the colon fibers and ground substance of this layer are mucins creating olfactory glands, including many blood vessels and nerves. Gustation is also known as the sense of taste. It permits us to perceive characteristics of what we eat and drink. The gustatory taste cells are also known as taste buds on the tongue surface. Papillae and taste buds of the tongue are broken into 4 types of papillae: filiform, fungiform, velatile, and foliate. Filiform papillae are short and bristle-like. They are distributed on the anterior 2/3 of the tongue surface. These papillae do not have taste buds and thus have no role in gustation. Instead, their bristle-like structures serve as a mechanical function. They assist in detecting texture and manipulating food. Fungiform papillae are block-like projections primarily located on the tip and sides of the tongue. Each contains only a few taste buds. Velatile papillae, or known as circumvelatile papillae, are the least numerous in prevalence, about 10 to 12, yet are the largest type of papillae found on the tongue. They are arranged in an inverted V shape on the posterior dorsal surface of the tongue. Each papilla is surrounded by a deep, narrow depression and contain most of our taste buds. Foliate papillae extend as ridges on the posterior lateral sides of the tongue and are not well developed on the human tongue. They house only a few taste buds during infancy and early childhood. We now proceed to looking into the organ system and organism characteristics of taste and smell. Organ system level 1. Olfaction and gustation are first and foremost under the auspices of the nervous system. Sensory information is passed from chemoreceptors in a process called sensory transduction. The muscular system is used in conjunction with the respiratory system for inhalation by way of the diaphragm. The 4 muscles of mastication aid in breaking down food to create aqueous saliva or transient mix, to increase the receptivity of the taste buds. Olfaction and gustation play a crucial role in relation to the androcene system, which can produce cravings in the form of heightened sensory sensitivities in response to nutritional states. This means that the olfactory and gustatory senses play a crucial role for the digestive system and to some extent the reproductive system. For the organismal level, olfaction and gustation have a prominent role in the human organism from a young age, largely aiding in development. Chemical senses are particularly sensitive in babies who use their sense of smell and taste to determine who people are. The loss of smell and taste can happen from various factors. Some of these factors are people are born with a weakened sense of smell and taste that could deteriorate. Infections or injuries can diminish your senses for a time. As you age over time, your sense of smell and taste diminishes. Disorders affecting smell and taste are actually very common. Approximately 2 million people in the United States suffer from this. Some diseases that can affect this are diabetes, Alzheimer's, Parkinson's, for example. This image picturizes the information that is shown above. Flavor is more than just a taste sensation, as often recognized by smell too. Sweet, sour, bitterness, and salty are 4 of the most commonly identified taste sensations that are also identified by smell. Familiar flavors are largely sensed and remembered by smell. For example, the smell of your favorite coffee produces a subsequent reminder of its taste. And that concludes my presentation. Thank you for listening and watching. I do have questions that were assigned to us. The first question that I'd like to ask is, why do the older people always complain about their tasteless meals? The second question I have is If you can't smell, can you taste?